1950, a new research tool was introduced to science. It wasn't a microscope or a lab technique. It was a mouse. A mouse with a genetic mutation that caused it to have severe obesity was now available to scientists. This creature, called OB, changed the course of fat research. It had unstoppable eating behavior that caused it to weigh 3 times as much as a normal mouse and have 5 times as much fat, leading ultimately to diabetes. In this mouse, researchers had a living system of obesity on which to experiment. The scientific name of the mice is OB/OB. Genetic nomenclature uses italics with the gene name given twice to denote that both copies of the gene have defects. For simplicity, the gene name is used only once in this book. The second development was an unforeseen leap in a different field of science. In 1957, the Soviet Union surprised the world with the launch of Sputnik, the first Earth satellite. This significant breakthrough precipitated a race for technological advancement. In reaction, the United States and other countries exponentially increased investment into scientific research. A portion of the increased funding was directed into the development of new scientific tools. Advances were made in biological separation techniques, such as gel electrophoresis and high-pressure liquid chromatography. Both methods allowed the separation of cell components and the identification of cellular proteins. Now researchers had means other than a microscope with which to examine cell contents. These tools, combined with the living system of the OB mouse, created many new research avenues. No longer confined to examining isolated cells under a microscope, researchers could now use the mouse to monitor the activity of living fat tissue and its impact on other organs. Scientists were able to more fully characterize enzymes in adipose tissue, the movement of proteins into and out of fat cells, and gain a clearer understanding of fat metabolism. Suddenly, there was a lot of talk about fat. Scientific periodicals dedicated to the subject were established, such as the Journal of Lipid Research. Although the understanding of fat was growing rapidly, the most astounding insights were yet to come. In a 22-year period, from 1973 to 1995, 2 scientists from different countries, spanning different generations, revealed how little we actually knew about the organ we love to loathe. Something in the blood. The first breakthrough discovery came from the Jackson Laboratory in Bar Harbor, Maine. Jackson is a breeding ground, literally, for world-class animal models of disease. Hundreds of mouse breeds are created there to embody human disease states such as cancer, Alzheimer's, and diabetes. Scientists study these mice to gain deeper insight into diseases. The OB mouse that revolutionized fat research had first been identified at the Jackson Laboratory. Douglas Coleman was a scientist at Jackson from 1958 to 1991. He had a friendly face made larger by a receding hairline and oversized glasses. Coleman grew up in Canada and developed an early interest in science, eventually attending McMaster University. He went on to the University of Wisconsin, where he earned a Ph.D. in biochemistry in 1958. He had planned to return to Canada after graduating, but the prospects for work there weren't promising. Instead, he took a job at the Jackson Laboratory. His plan was to remain for only a year or two, but as Coleman, who died in 2014, recalled, the laboratory provided a very fertile environment with excellent colleagues and world-class mouse models of disease, and I spent my entire career in Bar Harbor. I never dreamed that I would work on obesity and diabetes. One day in 1965, a researcher asked for Coleman's help to characterize a new strain of obese mice that had just been bred in the lab. These mice, called DB, were different from the OB mice. Not only were they obese, but they had a more severe form of diabetes. Coleman studied the mice for weeks and developed a hunch. There must be something in the blood of the DB mouse that was causing the disease to intensify. He arranged an experiment that would transfer the blood of a DB mouse into an OB mouse to observe the changes that would result. Using a technique in physiology called parabiosis, he sutured together sections of the 2 animals' tissues that enabled their blood to be exchanged. If there was something in the blood of the DB mouse causing severe diabetes, then the OB mouse should display the same symptoms. After the delicate task of surgically preparing the mice, Coleman was looking forward to seeing the experiment's results. The outcome, however, was drastically different from what he had anticipated. Once the blood flows of the 2 mice were connected and the OB mouse